Hi everybody, and thanks for watching today's interview. No More Excuses provides exclusive content for you, the ALS patient. We focus on real treatments. We focus on the today and the now, not what could be in another 5 or 10 years. 2 years ago, we identified TREGs as one of the 3 leading therapies that we would fight for because, number 1, it could be done, and number 2, it works. And as a patient, when I say it works, what I mean is it provides real hope. It provides hope that we could stop the progression of ALS. In fact, there are now 2 such therapies that have done this. One of them, Monday, sadly we learned that the FDA was not going to allow to continue despite clear and convincing evidence that it should. We have serious problems in ALS. It's terrible that as we start the year 2021, patients are dying right now because of lack of access to these drugs, not because they don't exist. So today we talked to Dr. Howard Berman of COYA Therapeutics, the new running back, if you will, for the ALS therapy called TREGs, developed by Dr. Stanley Appel in Houston, Texas. Please enjoy, and I'll be back for some very quick comments after the interview. Thank you. All right, ladies and gentlemen, with that, I'd like to bring in now our guest today, Dr. Howard Berman, all the way from Canada, where he is right now. And this is the CEO of a great, exciting new company called COYA Therapeutics. Dr. Berman, how are you today? And thank you so much for joining our interview today with No More Excuses.
No, it's a pleasure to be here. I've listened to your podcast, or I've listened to your prior interviews, and it was very impressive to me. And your passion really showed through, and I think all your listeners and supporters. And the way that you spoke about the regulatory T-cell therapy was really, it was symbolic of the way we feel in terms of its importance and potential. So it's a pleasure to be here, and happy to answer any questions you have.
Great. Yeah, the ALS patient community has been so excited about the TREG procedure. When we started No More Excuses, Doctor, 2 years ago, what we did is to essentially try to pick, if you will, the 3 leading therapies that we believed had the highest probability of success at the earliest time. We became about here and now, not the maybe 5 to 10 years from now. And one of those therapies was, of course, from an amazing man down in Houston, Texas, at Methodist down there, Dr. Stanley J. Appel. And that procedure to me was exciting because of, frankly, of the phase 1 results that had happened, and because of the way that this procedure seemingly has the ability to really slow or perhaps even halt ALS in some people. What was it, Doctor, about this procedure? And we're going to hear from Dr. Appel just in a moment about what he said about this procedure.
Let's fast forward, or not fast forward. Let's go back in time to earlier last year. I was at an appointment for my father at Houston Methodist. He unfortunately had a neurocognitive issue. It's not ALS, but we have all been struck by neurodegenerative disease. This is very personal for me. And because I've been many years in the industry, I've launched many drugs. I have experience with Alzheimer's and with neuro-oncology and immuno-oncology. The neurologist who my father was seeing suggested that I connect with Dr. Appel because of my experience and because what he knew what Dr. Appel was working on. So he set up a meeting for me to join Dr. Appel in a private session to go over Dr. Appel's regulatory T-cell breakthrough, his research, and see if there are any opportunities. And it was then in slide number 3, I believe, in which I was so captivated and taken aback on the accomplishments and the innovation of the approach. In my mind, I said, I'm going to leave AbbVie, where I was a very senior person at AbbVie, billion-dollar company, and I was going to take this on. So I resigned not long after that, and I said, I'm going to help Dr. Appel. I'm going to help you commercialize this. He asked me, you're a young man? I said, well, in some people's eyes, I'm a young man, but I want to help you. And I want to bring this to patients because it's such a devastating disease.
Wow.
And I truly believe, Mike, that we have something that is differentiated and unique here based on what we've seen in early clinical trials. And it appears to be very safe based on what we've seen so far.
I agree. Do you believe that TREGS now has the muscle and the commitment on your end to finally transform this therapy and get it into bodies and make it commercial?
I would not have taken this initiative on if I didn't believe that.
I think absolutely that is the case. And that's amazing that you came onto this so quickly after the third slide. But I think, why don't we just do this? Why don't we just play the infamous video of Dr. Appel himself so that some of our newer patients perhaps can understand the history a little bit of this drug and what kind of promise it may hold.
What was amazing was that we stopped progression of the clinical disease. At the very least, we had hoped that maybe we would slow the process. We actually stopped the progression of ALS in a way that had not been done before.
At a different age, but the approach we've taken is that neuroinflammation and the immune system may be a very important factor dictating rates of progression, onset, and survival.
Our goal is not to cure ALS. In order to cure ALS, one needs to modify the underlying insult at the motor level of the motor neuron, which in sporadic cases, it could be many genetic factors, misfolded proteins, and so forth. But the one common denominator, the lowest common denominator, or the most common feature is neuroinflammation. And it is the TREGs that are dysfunctional in all of these cases, whether it's sporadic or whether it's genetic. This is probably the best, in my opinion, opportunity to dampen that inflammation and to keep a sustainable and durable response. And we would like to take a similar analogy to HIV and AIDS. It was a deadly disease. Patients were dying, and the combination of antivirals and now has extended life for many, many, many years, and people live with it. That's what we would like to do with ALS.
Absolutely, no question. And that was part of our original strategy. Was really the first step is to put out the fire. You don't call the arson investigator first, doctor. You put out the fire, you go back and then find the cause of that fire. Is that a fair analogy in your case, and is that kind of what you're going after?
It's so true. Let me give you an idea as to what occurs. The motor neuron is initially based on the pathology in the motor neuron, the insult, whether it's a SOD protein or whatever it may be, autophagy, the cellular machinery is not working properly. What occurs is that the immune system sees that something's going on with the neuron. So it sends macrophages, but what we call good guy macrophages, they're M2 macrophages, that release cytokines to try and relieve that damage and that insult. But it realizes that you just can't repair it, and the motor neuron is essentially frustrated, and it ultimately sends out a signal that says, you know what, you didn't repair me, take me out. So the immune system becomes hyperactive, and it sends out bad guy macrophages called M1s and other effector T cells, and those are essentially what take out the motor neurons, and the motor neurons die. And what we're doing is we are blocking that forest fire, that cascade that occurs with the immune system. And we're not doing it through just one pathway. Every other approach, the complement inhibitors and all these other TNF inhibitors, they are impacting one pathway in the immune system. They all believe neuroinflammation is important. The problem with that is that there is redundancy in the immune system. The TREGs, by putting in millions upon millions, of cells into the patient of these good guy Tregs, they are the master regulator of the immune system. And by working upstream of everything else, they affect every downstream pathway and cascade. And that's why we believe we're going to have a significantly much more potent effect in blocking that forest fire.
A great explanation of how this works. I'd like to show everybody this interesting article that, frankly, came out around, I think, 2017, 18, somewhere in this time frame. This was the article that when I saw it, when I was first diagnosed, of course, it was so amazing to me. And it's Larry Bradley, Sharon Bradley, his wife. It says, quote, both times it stopped the progression of ALS in his ALS. He lost no muscle strength in his legs, arms and hands, and no lost breathing function during the period that he received the infusions.
They did remove his blood, pulled out those Tregs, multiplied them, put them back in to protect the motor neuron, and we saw progression slow. Look, at the end of the day, progression stopped.
This is not a cure, and patients are fine with that. I think the idea here that we need a cure at this moment, it would be fantastic, of course, and we hope that some of this new research, perhaps from even Dr. Osdinler's work, maybe at Northwestern, will regenerate motor neurons and be able to refresh them. But if we stop progression of ALS, as Dr. Appel correctly says, in theory, we kind of have a functional cure if given early enough in the disease.
Yep.
And I think the term functional cure is applicable here. Do you have any comments on that?
I think that the innovation that is going now with these folks and researchers that are working on regeneration of motor neurons and other mechanisms to affect the underlying disease process is exciting, but it's many years away for seeing the light of day.
Absolutely.
And what we would like to do is to have patients live with no change until that time, until they can be, those neurons can be repaired. And we want to stop progression, ideally. Absolutely. So yes, that's what we're aiming to do here. The phase 1 trial showed in a fast progressing, a medium progressing, and a slow progressing patient, when you gave the infusions, it stopped. It didn't delay slow progression, it stopped progression. Dramatic and remarkable, and it did it twice. In the first series of infusions, then they stopped the infusions, and guess what happened? Patients eventually started to decline again. But then they reinfused the cells, and guess what happened? The patients stopped progression even later on when they had further deterioration. So now we're conducting a phase 2A trial, Mike, and we've converted fresh cells, which you can only give, once or twice a year, because it's very cumbersome to do leukapheresis. Now we've perfected cryopreservation.
Wow.
And we're able to make billions of cells on the first manufacturing run. We're able to aliquot the cells, and we're able to rethaw them with full functional activity. The problem with regulatory T cells, Tregs, is that they are very different than CAR T cells that they're used in cancer. They are very unstable, they're finicky, and they die very easily. So no one had been able to cryopreserve them until we were able to do it. That's part of our intellectual property. It's part of what we do, our manufacturing. So one leukapheresis done once a year has enough cells for the rest of the year for the patients.
Absolutely.
Infused monthly so that the responses can be sustained. And that's part of our phase 2A program, which will end in May of this year with a full data readout. Dr. Appel did a preliminary translational research in mouse models, and he crossed a specific kind of mouse model called a SOD mouse model with another mouse that didn't have an immune system. And the thought was that if you actually take out the you take out the immune system, you take out the T cells, then the mice would live longer because you don't have that inflammation and that inflammatory effect. But guess what? The mice lived shorter. And the reason is you didn't just take out the effector T cells, but you took out the regulatory T cells. So then he added back the regulatory T cells, and guess what happened? The mice lived even longer. So it proved that regulatory T cells are very important. Then he decided he took another step and he took blood from patients, ALS patients, many of them, and he found that the regulatory T cells were dysfunctional. Sometimes they were 15, sometimes they were 20% of where they should be. They were dysfunctional and they were lower in number. So he realized, and he also did some experimental work, and he correlated that the degree of dysfunction correlated to the survival. So if patients were dysfunctional to a very strong degree, they had shorter survival. So all of these correlations and these links led to the idea, let's do a phase 1 trial and let's take these patient cells, we can expand the cells and we can convert these cells from being dysfunctional to functional. And he was able to receive funding from a very wealthy benefactor who had a link to ALS and run this phase 1. And he ran it in 3 types of patients, a fast, a medium, and a slow, because he wanted to do it in each category.
Of course.
And the results just were beautiful. It proved to him that the regulatory T cells were going to be effective, at least during the time course of the infusion. We have learned subsequently in the phase 2A, which part of it is open label, part of it's double blind, we have learned a significant amount more from the phase 1 as well. And, I'm happy. I don't want to get too much into the phase 2A until it's unblinded, but yeah, I think we need to be we have to use the scientific method and look at the data and analyze it, and then we can release it to the public.
That's incredible. And that's pretty much matches kind of what we had thought up to this point, the path that this drug was following, or this therapy was following. Please defer if you need to. Recently, with the sad, tragic defeat of Neuron, are you hopeful that, and are you aware of what is happening to make sure that this kind of thing can never happen to TREGs? Because we can't afford to lose another product like this. Would you care as much as you can to comment on steps that you may be taking, or are you aware of this issue?
Very aware. And first, let me say that I think that what Brainstorm was doing and is doing is very exciting. I think they, have an exciting platform. I don't know the minutia and the details of what's occurring or what the rationale for the decision with FDA. I can't comment on that, but certainly it seemed that they were having an influence and an impact on biomarkers, which are critical in inflammation, and certainly probably based on what I've read is that there were subgroups who really benefited from treatment. I will say in terms of what we're doing, we have very specific endpoints and biomarkers that we're looking at. And, we are looking across the spectrum of inflammatory response. We are looking at TREG suppressive function and a whole slew of other markers. And we're using different scales, and we're using both a standardized ALS-FRS as well as another scale that is very strong in terms of, it's actually Dr. Appel's scale that he uses. And it's it's a very objective scale that is used to assess response. We are also, we're looking very, very carefully at stratification, but, our goal with our Treg function is to not leave anyone behind, per se. We would like to be able to treat all patients. We wouldn't particularly like to stratify by fast progressors or medium progressors, and we don't want to specify by or stratify necessarily by genetic predisposition or genetic ALS versus sporadic ALS. And so when the data becomes very clear to us and we read out our data, we will have a lot in front of us in order to ensure that the next trial that we run with consultation with the FDA, of course, is run so that we have the best chance of success to bring this to patients ultimately.
Okay.
And I have to say, I know it's very disheartening for patients to be in a trial, and for those who receive benefit to not be able to ultimately have to leverage the therapy in the future. I know it's frustrating, and like I said, I have a personal history with family members, and you really want to do everything you can to see benefit to your family members. So I feel very sad for patients and their families, and we just want to do everything we can to benefit and bring a proper therapy to patients.
Thank you for those words, and I couldn't agree more. I believe, in fact, this is about building a multidisciplinary approach, I believe, like you mentioned earlier with HIV. Perhaps some versions of ALS, maybe they don't have an anti or an inflammatory involvement. Maybe theirs is more of a mitochondrial or TDP-43 pathway issue. We don't understand that. So there's probably different versions of ALS, which means, of course, as we all know, it's tremendously heterogeneous, which makes the idea of a homogeneous therapy nearly impossible, frankly. And that was what was disappointing. Go ahead.
Let me state this, that I was going to say ALS is very heterogeneous, and there are patients even patients that are on placebo, that will do very well. And that, I think, is one of the biggest confounding feature in ALS trials. You have to power your study. In other words, you have to have enough sample size in order to weed out a placebo effect. And number two is the response. So if you're only benefiting patients or reducing decline in patients by a certain percentage, then you have a better chance of having this placebo effect. But if you are stabilizing ALS and the patients aren't declining, right? It's hard to get that placebo variable that will confound the data. So the data will actually prove out whether the trial is successful or not. And so we are hoping that our regulatory T-cell therapy can stand up on its own in terms of the clinical data, and really show even the naysayers that this is something that can be taken on its own face value as something that can be beneficial and not impacted as much by this placebo effect.
Many times patients ask me, of course, I'm not a doctor, I'm not a medical doctor at all. They ask me what are my thoughts about Radicava and, of course, drugs like this. And there's been this idea in ALS of drugs that can help by 3 to 6 months. And that's really not what patients are looking for. We're looking for, as you said, indisputable evidence, if you will. If somebody halts progression or stabilizes, for instance, it's much easier to read that effect in a trial clearly. The only problem is that not everybody in that trial will stabilize. And for some reason, that doesn't seem to be good enough in the ALS kind of scientific community, I guess, if you will. And that's a real shame. And I think what we've got to do is just learn to read the noise a little better in these trials, because there's always going to be noise in ALS trials. And we've got to make sure that we don't add good drugs to the list of drugs that have failed, which I think is now over 80, I believe. It's somewhere in the 85 to 88 failed trials.
I'll tell you this, Mike, that, like I said, we have learned a tremendous amount. And we have some very exciting information and data. I think we also have, and Dr. Appel is working on lots of proteomic data, which will also be able to help stratify out responses and whatnot. I mean, this is, many multiplex proteomics, which looks at, dozens upon dozens of proteins that are all impactful in the inflammatory response. So stay tuned and stay tuned till the summer. And I think I'm hoping that we're able to share this with you.
Hopefully they'll be good, and we expect it. I'm praying and hoping that they will be. The last question I have is, before we go, I know you've got time is short and you've got to go. Tell me a little, because everybody's asking this question now, timeline. Talk timeline for us. What is the plan for the next part of commercialization, if you will?
So we are right now looking to scale up our manufacturing. One of the things in cell therapy is that it's great to be able to do it, but can you industrialize it? So we are in a scale-up GMP and CMC, which is the manufacturing component of our regulatory T-cell approach. we want to be able to provide this in large numbers and show the FDA that we can do it. So we are planning, we're currently in the process of designing and planning our next trial, which is going to have a larger number of patients.
Okay.
And that trial will be, hopefully, we're aiming for perhaps the end of the year, ideally, but stay tuned on that.
Okay.
And like I said, we have strategies and approaches which we've learned where we're going to be taking our regulatory T cells and we're going to be combining the regulatory T cells with other things, which we've found to be synergistic and can even likely help and improve the response as versus just the regulatory T cells by itself.
Okay. So phase 3 trial, hopefully, let's say end of fourth quarter 21, maybe early, first quarter 22, possibly. That's kind of the plan.
We likely will do another phase 2, which
Okay.
may then lead into a more detailed registrational trial. But we'll probably go through another phase 2 to learn a few more items.
I thank you so much for speaking with us today. You're talking to patients right now, and that is huge, and I very much applaud you for doing that. And I just want to say on their behalf, thank you so much. And it sounds like TREGS, as we know it, is in great hands. It really is. Anything you'd like to leave with? And I thank you so much.
I would. I'd like to talk to the patient community. As Dr. Appel says, ALS is a disease. He calls it nice guy's disease. But it can also be nice guy and nice gal's disease because women have ALS as well. And I would like to say, most importantly to them, that we've got your back. We will do everything in our power at COYA to bring this therapy as quickly, as safely to patients. We want to ease the pain and ease the burden on patients and their families, their caregivers. This is a mission for us. It's a personal mission for me, just on a humanistic level. I really feel for patients, and I just want to do the best here.
Amazing. Thank you so much. And I guess the very last thing I'll leave with is, what about donations? How can patients continue to support this? Being commercialized now with Dr. Appel, has that changed the ability for patients to go and to donate to this therapy? How would you prefer that they handle that?
At this point, I probably would hold off on donations. We've got the money that we need, which is good news, in order to bring this forward. But I mean, certainly we can explore and revisit that. But, patients' money, I would like patients and their families to keep their money, for their own needs and for their care. And God willing, hopefully we will soon have even additional capital and funding so that we can bring this to more patients with larger patient trials.
Amazing. Thank you again so much. I look forward to hearing hopefully, we'll keep our fingers crossed, a good readout. Please come back anytime you'd like to speak to the patient community, and we eagerly await those results. Dr. Berman, thank you so much, and we're watching very closely, and if there's anything we can do as patients, let us know. We'll be happy to help.
Mike, thank you for what you do. You're on a mission, and I very much appreciate what you're doing. And I think that you're really contributing and making a step forward for yourself and for the community. Thank you.
Thank you so much. Goodbye. Thank you so much to COYA and for Dr. Berman for doing this interview today and speaking, frankly, directly to the ALS patient community. It's critical that as we keep doing these things and as we keep hope alive for ALS patients who are alive now, that means you or your loved one, that we understand that our system in ALS is flawed. The drugs exist to stop this disease, and I think that's something that more ALS patients need to be told and need to understand. That's our mission here. Very simply, is to put and help get drugs in bodies. That's it. Because at the end of the day, that's what really matters to an ALS patient. I salute Dr. Berman for coming forward and talking directly to the patient community today. And we're going to keep doing more of these things because we need the word to be out. Thank you for watching today. I'd like to thank my guest again, Dr. Berman from COYA Therapeutics. We'll see you all later. Thanks.